I can already start thanking Edor for the nice introduction and also for the invitation to give a talk here. I must say that I was a bit in shock when I saw the program, especially because of the title in French. So I took the freedom to change it a little bit, and so, well, it's still, I think, the same content. But yeah, it's now in English, and I hope that that's no problem. By the way, I understand French, so be careful. So I don't have to introduce ALS for you, I suppose. And what we want to do here is to continue research on ALS using patient cells. Therefore, we isolate fibroblasts, skin cells from patients. We convert them to iPSC cells, induced pluripotent stem cells. And the nice thing is that we can also correct the mutations in these cells. And then we can differentiate these cells in different cell types. And that's the nice thing about the whole system. And for the talk of today, of course, the most important cell type we're focusing on are the motor neurons. We were very lucky when we started this project that we had collaborations with people in France. The protocol here is the Maury et al. Protocol, which was developed, I think, in the lab of Professor Martina. And we were also lucky that Delphine Boel, working here, helped us to set up this protocol before it was even published. It just reproduces, in a very complicated way, the embryonic development. And this ends after, let's say, 1 month with cells that look like motor neurons. And that also show the markers that you expect from motor neurons. What you see here are a few stainings of motor neurons that were obtained from iPSCs that came from patients with mutations in FUS. You can nicely see the neurites. You can also nicely see that the markers that we have used, is islet-1, cholinacetyltransferase, that they all show nice stainings. And what is very nice about this protocol, and I think, well, that's my personal opinion, that it's the best protocol around, that is that almost all cells are neurons, and 80 to 90% of the cells in the dish stain with the typical motor neuron markers. What is already important to mention here is, and it's something we have seen for all the other iPSC lines as well, that is that there is no difference in the differentiation efficiency when we start from patient cells or from controls. What is also nice to see is that these neurons, these motor neurons, are electrically active. And also there, both the motor neurons from patients and from controls are electrically active. There are some differences there, but, well, nothing spectacular, I would say. What is also interesting to see, and, well, we're working here with FUS, is that what we see in patients also is seen in these relatively young motor neurons. You see here the mislocalization, for instance, of FUS. You have to focus on the green. So normally FUS is a nuclear protein, and then you can see in the two patient lines with the two different mutations that FUS suddenly is also visible in the neurites. So what is seen in patient reproduces also in culture, always nice as a starting point. What is also nice to see is that indeed the axonal transport, which is very important in motor neurons, and I will come back to that in a minute, that is also happening in these cultured motor neurons. What you see here, I hope you can see it, that is that the mitochondria, because we have loaded the cells with mitotracker, that the mitochondria are moving along the axons in both directions. And when we studied the axonal transport properties in the different lines, it became very obvious that over time, axonal transport became compromised in the mutant lines, in this case, in the FUS line. You can already see it in the chymographs on the left. On the right, you can see the quantification. You see that initially at week 2, everything is still more or less okay, but then over time, the movement, the number of moving mitochondria is going down significantly. It's not something that is typical for mitochondria, so it's a general axonal transport problem because when we load these cells with ER, a marker for ER vesicles, or well, when we just check the lysosomes or even RNA, then we see the same. And what is illustrated here is for ER. You see for two different mutations that indeed we also see here that same difference that I've just shown you for the mitochondria. I don't think I have to introduce motor neurons to you, but well, I like this scheme, especially because it reminds us that motor neurons are mostly axon and only a very small cell body and a little bit of neuromuscular junction. And so, yeah, well, that means that axonal transport is even more important in a neuron that can have an axon of more than 1 meter than in any other neuron. With having these data, well, there is another thing, of course. When you want to publish these days results with iPSCs, then, well, the referees always ask for isogenic controls. I've told you in the beginning that this is one of the other advantages of iPSCs. And indeed, what we have done in this case is we have corrected the mutation in FUS. And what you can see here, that FUS, and now FUS is stained in red, that FUS goes back into the nucleus. And that we can also rescue the axonal transport defect, as is shown on the right. Which means that indeed that point mutation in FUS is responsible for the axonal transport defect that we observe. Is this unique for FUS? No, it isn't. In all the different iPSC-derived motor neurons that we have been studying until now from ALS patients with familial form of ALS, we see something similar. Whether it's FUS, what I've just shown you, whether it's mutations in TAR-DPP, encoding for TDP43, or whether it are lines with these repeats in C9orf72, as is shown here on the right. These are the chymographs. This is the quantification. This I've already shown you. TAR-DPP, same story, lower number of moving mitochondria in the different lines. And then also for C9orf72, this is the most recent. These are the most recent data. In green, the green bars are control lines, and then you see in what's the color pink, you see the C9 lines, and then in blue, it's the isogenic control where the repeat is removed from the line, and you can see that indeed, when the repeats are present, there is less axonal transport. When the repeat is removed or in control lines, there is more. Okay. So that seems to be a general thing, and that's always nice because we are looking for things that are not just specific for one type of patient or for one type of mutation, but something that might be more important in general. The next question, of course, is now that we have a defect, can we correct it? It's always nice that when something goes wrong, that you find a way to cure it or to restore it. And therefore, I just have to introduce you what the reason could be for that difference in axonal transport. And that is the fact that the microtubuli can become deacetylated. Acetylation is a modification of alpha-tubulin. When we have deacetylated microtubules, then the motor proteins don't attach very well to the microtubules. And so the acetylation status of the microtubules is very important for that. We also know which enzyme is responsible for the deacetylation of the alpha-tubulin, and that is HDAC6, histone deacetylase 6. It's a very misleading name because HDAC6 is deacetylating a number of substrates, but histones are not included in that list for the simple reason that HDAC6 is the only HDAC that is present in the cytoplasm and not in the nucleus like the other HDAC. So this has nothing to do with epigenetics. I'm still invited for meetings about epigenetics, and I always have to explain that we are not doing anything related to that. HDAC6 is a cytoplasmic enzyme and, well, deacetylates alpha-tubulin. We also have very good HDAC6 inhibitors, and therefore it's possible to reverse the deacetylated state of the microtubules and to have normal acetylated microtubules again. Here you can see that indeed these inhibitors do what they are expected to do when we incubate our cultures with ACY738 or tubastatin A, which are two selective HDAC6 inhibitors. You can see that the band of the acetylated alpha-tubulin becomes more intense. So indeed, it is doing what it is supposed to do. Yeah. I see that my videos are, well, not any longer in my presentation. So luckily, I've also included the quantification. So here you can see the effects of the inhibition of HDAC6 on mito tracker and on ER tracker. And so you can see in control conditions, the blue conditions, there you don't see any effect of the inhibitor. So there, apparently, the acetylation level is already maximal. But then when you treat the mutant lines with an HDAC6 inhibitor, and we do that just overnight, so it's just an overnight treatment with an HDAC6 inhibitor, you can see that both in FUS and in TDP43, that indeed the problem is solved, so to say, that the number of moving particles is again, well, at the same level as in the controls. And that both for FUS and for TDP. So that brings me already to my first conclusions. So what we believe is that mutant FUS and mutant TDP43, in one way or another, and we can discuss about that they cause axonal transport defects, that HDAC6 inhibitors, by changing deacetylated alpha-tubulin into acetylated alpha-tubulin, that they, that they reverse these problems and that we have normal axonal transport. And I've also shown you that isogenic correction of mutations can have the same effect. At least I've shown it for FUS, but we also have data for TDP43. So whenever you find this data, your institute wants to send a press release out. And in this case, the title of the press release was something like traffic jams are causative for axonal transport problems in ALS, something like that. And then, of course, when you think about the traffic jam, and by the way, most of these data were obtained by a Chinese PhD student. So then you think about situations like this. Too many cars on the road and nothing is moving anymore. And so my student came to me and said, no, I don't think that that's what's happening. I said, yeah, but well, you don't have to make it too complicated. Everybody knows what a traffic jam is. Everybody understands that if we can solve traffic jams, that more people will be happy, not only with ALS, by the way. I said, yeah, but come up with another example. Yeah, well, can you do better? And she came up with the next slide, and I still like it, so I'm going to show it. She said, what's happening is actually that there are not too many cars, but that the road is damaged. So mutant FUS and mutant TDP, in one way or another, damage the microtubules. And by inhibiting HDAC6, we solve that problem and we have a good road again. So the microtubules become acetylated and the cars are moving again. By the way, here it looks as if the cars are damaged. We have looked at the mitochondria. The mitochondria are healthy. They function normally. So it is just the road that is damaged, and it's that what we can solve with the HDAC6 inhibitors. I've told you this story for FUS and TDP. Of course, yeah, we have seen that also for the hexanucleotide repeats in C9orf. It also works. There we can indeed restore axonal transport by removal of the repeats, but also by inhibiting the HDAC6 inhibitors. These are the most recent data that we have. Don't ask me how it works. We are investigating this at the moment because there must be another explanation for this when we compare it to FUS and TDP. But here you can see again that the axonal transport problem that we see in C9 is restored by an overnight treatment with tubastatin A. Okay. This was actually the story I wanted to share with you today, but I want to go one step further because at the same time in the lab, we are not happy anymore with just isolated motor neurons. We know that other cell types also play a role in ALS. So the question that we asked us a few years ago was, can we increase the complexity of the system? And so what then the idea is to bring all these different cell types together that we make from the iPSCs and that we recreate what is called a spinal cord on a chip or in a dish, eventually combined with some electrodes. And we worked with the axonal microfluidics, commercially available microfluidics that are composed of two compartments, the green and the red one, and they are connected to each other with very small channels. What we do is we seed the motor neurons on one side, we seed muscle cells, myotubes in the other compartment, and then we put a gradient, a growth factor gradient over the channels, and then the axons grow into the direction of the myotubes, and they make contact with these myotubes. At least that's what you hope. The good news is that it also happens in reality. What you can see here is one of the first pictures, and I still like it very much. On the left, you can see the motor neuron compartment. In blue, you see the nuclei. Green is neurofilament. And so what you can see here is that the axons, and these are multiple axons, that they cross these channels, and they are 900 micrometers long, and that they go into the direction of what you see in magenta are the myotubes. And indeed, they grow into the direction of these myotubes, and they make contact. This is also nicely illustrated here, where we have combined the neurofilament staining with a bungarotoxin staining. And on the right, you can see on an EM image how the contact points look like between the myotubes and the axons. And you can also see that they try to make the surface larger, what is also seen in a normal neuromuscular junction. The nicest picture I've saved for last, so this is also another one. So these are the myotubes, and there are two axons coming from both sides, and they seem to like the muscle cells very much. Okay. Of course, it's not because two cells make contact that they also communicate with each other. And therefore, what we did was we stimulated the motor neurons on the left side, and we recorded calcium transients on the right. And indeed, what you can see is that indeed we can induce calcium transients in these cells. And, well, this is information that is going over the neuromuscular junction, because if we add curare to the system, then we don't see any calcium transients anymore. So it's really proving that these are functional neuromuscular junctions. Okay, now we have a nice system. Now, of course, we can start doing the most interesting experiments. So what can we do with these neuromuscular junctions? First of all, we can see what the effect is of having mutations in the motor neurons. And indeed, what we see is that, and this is FUS again, that over time, the number of neuromuscular junctions goes down. So initially, when you see in red, the number of neuromuscular junctions that are formed is more or less the same. But if we wait 1 week longer, in the control conditions, it's still at the same level, but in the FUS condition, where there are FUS mutations, it's going down. So ALS neuromuscular junctions seem to be lost over time when there are mutations present. So that is interesting. And that reminds us of what is happening in patients where, well, you know that the dying back hypothesis is still a very popular one. So this is the normal situation. And what you see, at least initially in a patient, is that the axons detach. And that seems also to be the case in our simple culture system. So we also have some kind of denervation that is happening not in a patient, but in a dish. What else can we do? We can start to add extra cells. So what have we done already? We're doing many different things, but well, we also have added astrocytes. We have a good protocol to make astrocytes from iPSCs. And here you can see the astrocytes stained with GFAP that are in between the axons. And we have added these astrocytes to the motor neuron compartment, where they normally also are in the spinal cord. And then we have checked what the effect is of astrocytes on the number of neuromuscular junction at the other side, in the muscle compartment. It's a lot of work, but this is the result, or this is one of the results. And what we see is, I'm going to guide you through this slide, is that when we add mutant astrocytes to a control motor neuron that has no mutation, then the number of neuromuscular junction is going down. So having mutant astrocytes is not a good thing. So the number of neuromuscular junctions are affected by the presence of that mutation in the astrocytes. And the opposite is also true. When we add control astrocytes to mutant motor neurons, then the number of neuromuscular junction increases. So astrocytes have a double function. They can have a negative function when they have a mutation, but when they don't have a mutation, then they help mutant motor neurons to make more neuromuscular junction. And now, of course, we are trying to figure out which pathways are involved in these two processes. But that's for next year, maybe. What else can we do? We can start to use this system to test therapies. And I've already introduced you HDAC6 inhibitors, so I'm not going to make it too complicated. What we see is at a certain stage of the development, that is that the axons that come out in the muscle compartment, that they grow slower when there is a mutation present in the motor neurons. When we treat these cultures with an HDAC6 inhibitor, the one that restored axonal transport, then we also see that suddenly the axons grow much better. And by the way, also the number of neuromuscular junctions are better, and they are also more complex. Okay, so that is now what we are trying to do. I'm just gonna, add one more advertisement. I have a company started 2 years ago, just before COVID, which has as main aim to develop HDAC6 inhibitors, improved HDAC6 inhibitors, not actually in the first place for ALS, but especially for peripheral neuropathies. But of course, what we hope with all the results that I've just shown you, that these inhibitors might also have a role in ALS. Okay, I come to my conclusions. I've just focused on two aspects in the interest of time, on the axonal transport defects and on the defects in the neuromuscular junction formation. We have, and I think that's a nice achievement in a relatively simple system, we have functional human neuromuscular junctions. I've shown you that astrocytes can influence the phenotypes both in positive and in negative ways. And, if you don't know by now that HDAC6 inhibitors can do something important, then, then you were not paying any attention to my talk, I suppose. Last but not least, I would like to summarize what I've just shown you, that I think that the system that we have now, the spinal cord on a chip, that could indeed not only lead to mechanistic insights, but hopefully also to new therapeutic targets and therapies, and that hopefully can help ALS patients. And then, of course, I have to thank the people that have done all the work. I didn't do any of the experiments that I've just shown you. I'm so lucky that I have a very talented and big lab that, still likes me, apparently. Also, there are a lot of external collaborations. There are funders that help us. I would like to especially mention the ALS League, the patient organization from Belgium that is supporting our research. And then, last but not least, I would like to thank you for your attention. Thank you very much.
Thank you, Ludo, for this very nice opening talk. Anybody has a question?
I would like to get questions from PhD students, by the way.
Very nice, Ludo. Congrats. So, is this working? Do you hear me?
Yeah.
Okay.
Loud and clear.
You showed these data from the astrocytes co-cultured in the multi-chamber system with the motor neurons, and so the mutant astrocytes had a detrimental effect on normal motor neurons, whereas normal astrocytes had a positive effect on the mutant motor neurons, which fits with data from other experimental systems.
Absolutely.
Okay, from SALT and so on.
Yep, indeed.
So my question, since is the effect of the astrocytes is probably via the motor neuron cell bodies or the proximal axons, right?
Yeah.
Have you controlled for this? Is the effect that you see on the neuromuscular junction specific? I mean, are the motor neuron cell bodies still all there?
Yep.
Or not? Have you counted the motor neuron? Or is it an early effect at the junction which later translate in effects on the cell body?
It's a very good question. And I forgot to tell you that in none of our conditions we see cell death. So whether or not we have mutations, well, that's of course always a criticism you get when you try to publish data like this, that they say, oh, yeah, well, indeed, ALS is a motor neuron disease, and in the end, motor neurons die. I think the culture conditions that we offer to these cells are so good that motor neurons don't die. And so, yeah, then indeed, the effect that the astrocytes have is not on the number of motor neurons. That stays the same. And yeah, the most interesting, and I hope that was clear, is that the effect in the motor neuron compartment translates to something that happens 900 micrometer further away in the other compartment, which is also what you see in patients, where there is also the place where the astrocytes are and where the neuromuscular junctions are. Yeah, there's 1 meter in between, if you're a bit larger. Okay, yeah.
It's remote protection.
Yeah, which makes it...
I have one question. Do you think that is it a key event that may occur very early in the phase, and so at the pre-symptomatic phase of the disease?
Yeah, that's what we hope, of course. I mean, it's difficult to prove, but my major fear, I must be very open on that, and I think from many other people as well that we're working with iPSCs, that is that, well, having such an embryonic system, that nothing goes wrong, that these cells, also because of the medium conditions, that they are happy and, well, live forever and perform all the things that they normally perform. So indeed, what I believe is that what we see in these culture systems could be the earlier things. And then, of course, and that's also, it's a chicken and egg problem, what, of course, we would like to see is what comes first, what comes second. And yeah, you want to treat something that comes very early. That's the whole thing. I mean, and that's, I think this system is a very nice one to investigate this. Yeah.
Thank you for the very exciting talk. I would like to know whether you have ideas about the mechanisms that would link first mutation or TDP43 mutation to axonal transport effects.
Oh yeah, I forgot one more. Okay, no, what I didn't tell you, and well, in the interest of time, I removed this slide, but yeah, if you ask me, I cannot, no, I have to mention this. I didn't tell you the whole story about HX6. HX6 is a deacetylase in the cytoplasm, and that's why it has, well, it has a nuclear export signal, so that's the reason. But it also has a very strong ubiquitin binding domain in the C-terminal part of the enzyme. And it's the one with the highest affinity in any other protein. So what we believe, and it's a hypothesis that we originally formulated in the context of peripheral neuropathies, that is that HX6, the role of HX6 is to remove ubiquitinated proteins from the axon. That's also the reason, I think, why it is not only working in models of ALS, but also in models of other diseases that affect the axons. And that, there's also, that I didn't mention, there's also a binding domain to motor proteins in between the two deacetylase domains. And so what is happening then is that ubiquitinated proteins are removed from the axons, where there is no proteasome, where there is no autophagy, and that they are then destroyed in the cell body. And that then what happens as some kind of an unforeseen side effect is that the enzyme is doing what it was supposed to do maybe earlier during life, and that it starts to deacetylate the microtubules, and that then induces the disease. And that then, of course, by inhibiting HX6, or at least by inhibiting the deacetylase function of HX6, that then, is beneficial. By the way, removing or deleting HX6 has not the same effect as inhibiting the deacetylating function, which is in line with what I just tried to explain. It's a hypothesis, but until now, most of the data are still in line with that hypothesis. Hi, Severine.
Ludo, thanks for your talk. Would like to come back to this experiment where you put the astrocytes. Mm-hmm. So basically here with the conditions you do, I assume that what you see is that you maintain the NMJ compared to when you put the control astrocytes compared to when you put the mutant one. Yeah. Can you play around with your system and actually see if you can get regeneration with control astrocytes?
Yeah, the problem is, of course, that you always have to fix these cells. So that makes it difficult to follow over time what is really happening. So you fix it at a certain moment, and then, yeah, well, you can do it twice or 3 times, but just following over time what exactly is what could give an answer to your question, that is a bit more difficult. That requires...
But if you put them later, actually, when you already have, because you see that in the mutant you have less NMJs, if you would put the astrocytes later, would you...
We didn't try. Yeah, well, and you could... Well, we are now adding microglia. We are playing around with Schwann cells. I mean, whatever, yeah, you can think of, we can do, but yeah, it's always... Well, it looks less work than it actually is, I can tell you.
Yes, very nice data. I have 2 questions. The first one concerns the Schwann cells. Did you try to complexify more by addition of Schwann cells?
We are doing these experiments. And then we focus mostly on the terminal Schwann cells, because what doesn't work is getting myelination of human sensory or motor neurons. For one reason or another, human Schwann cells don't want to remyelinate human neurons. When you take, for instance, primary rat or mouse neurons, then it works. But for reasons I don't understand, and I think most people that are working in that area have had the same experience, it doesn't work in a fully humanized system. But nowadays we are checking the effect of terminal Schwann cells, especially in the context, not so much of ALS, but of CMT, Charcot-Marie-Tooth disease, where there are indeed demyelinating diseases where the mutations are in the Schwann cells. That makes it interesting.
Okay. And my second question concerns the astrocytes. Did you try to put some conditioned medium from astrocytes directly?
Yeah, that doesn't have this. Well, that was what you also thought. Maybe we can just take conditioned medium, but that doesn't give the same effects. So I think the contact being there in proximity seems to be important. So just well, only the conditioned medium has not the same effects, unfortunately, I would say. But yeah.
We have a question.
Hi, Ludo. Here. Yes, I can.
Yeah, don't fight.
I'm here. Thank you, Ludo. I have a short question about the sporadic cases. Did you try motor neuron from these cases? And my second question is more on isogenic controls. Do you always see a complete correction of the phenotypes? As we know that there are some differences between the different mutations, etc. So we have no full penetrance. So do you have all phenotype corrected 100%?
No, not well. We didn't always check, I think. But for instance, for the axonal transport, I can be very clear. There, it's fully corrected. Do I know examples where it's not fully corrected? No, not as far as I remember. But I understand your point. Regarding your first question, I think that's the big unknown. Whether we can use this system for something that we could not study before, sporadic ALS, 90% of patients. We have no good models for that. I think a lot of people are struggling. The major problem there is that you cannot work with isogenic controls. So then you have to compare, let's say, 10 controls with 10 sporadic patients, which is not so easy. We have seen differences, I must say, but not in the same way as what I've shown you for the familial cases, for the simple reason that the variation is so high. So when we compare controls with sporadic ones, I mean, it's huge, the variation between the different lines. For instance, for axonal transport. We have seen, by the way, effects on axonal outgrowth, on branching, which seem to be significant. But yeah, it's easier said than done. And I write it in every grant application that I apply, that we now have finally a good model for sporadic ALS. I'm not so sure whether that is completely true. But I mean, yeah, if anything, it's a lot of work. Yeah.
And the last question.
Thank you. I had a question. I'm here.
Yeah. But I was looking there because somebody was looking angry at me.
Whether you have tested the contribution of the muscle to the neuromuscular junction formation in your system.
Yeah. Yeah, that's a good question. What I didn't tell you, that is that the muscle cells that we have for the moment are not iPSC derived. So these are satellite cells, mesangioblasts, that fuse much more efficiently, at least in our hands, than the iPSC-derived myotubes that some people can obtain. By the way, we can also obtain them, but, well, not in the same magnitude as what we would expect. And then, but it's something that is still on our list to check whether indeed mutations in the muscle cells have an effect. I would not be surprised, knowing that it's, of course, a very controversial thing in the field. I would not be surprised that mutations in the muscle cells in the system that we have would also have an effect. But we didn't test it yet for the reasons I just explained you.